Good afternoon, everyone. I hope you all are having fun and wonderful time here at the Symposium. I'm Aarti Singh, and I'm Associate Professor at Iowa State University. And today I'll be co-chairing this session on next-level AI models and algorithms for Phenomics with Germano Gomes from VIB Agro Incubator from Belgium. So in today's session, we have an exciting lineup of talks ranging from AI applications to data analytics to role of conversational AI agents and decision support to advance 3D reconstruction techniques. Without further ado, I would like to introduce our distinguished keynote speaker today, Dr. Baskar Ganapathysubramanian, who is Anderling Professor of Engineering at the Iowa State University. He's also Director of AI Institute for Resilient Agriculture at Iowa State University, and he has longest surname with 20 letters. In today's talk, he'll be talking on various AI-based tools for phenotyping, spanning from plant level to plot level to production scale farming. So please join me in welcoming Baskar to the stage.
Thanks, Aarti. Thanks, Aarti, for the introduction and stealing my thunder in the way I introduce myself is I have the longest last name, and when I teach my both undergrad and grad classes, the first pop quiz is what is your instructor's last name, and that always breaks the ice. So today I'll be talking a little bit about some of the work that my group, as well as a broad group of collaborators, have been working on in terms of building AI tools to enable a resilient agriculture system. And the two themes that I want to perhaps focus on, the key takeaways are the second line there, that as a community, if you collect data and we build these what we call foundation models, this can be easily fine-tuned for a variety of downstream applications. And this is the other theme that has been pervasive in the last couple of days of exciting talks is how can we leverage 3D traits? How can we extract our 3D traits and leverage them for both bidding and production? So I am privileged to lead a large institute called the AI Institute for Resilient Agriculture, which is funded by the National Science Foundation and the USDA and IFA. And so this we are a team of about 33 researchers, faculty researchers, working on between 5 and 10 year long problems. And the key idea here is can we advance foundational AI, fundamentals of the AI discipline through user-inspired applications in agriculture. And the focus is to looking at two agronomically relevant scales. One is the bidding scale, and the other is the production scale. And so our vision is given in the figure on the right, where we have 4 broad themes that we try to encompass in all our moonshots, all the projects that we are thinking about. Top left is how do you build these AI tools through, again, user-inspired lens, and then we deploy this at whatever scales, at whatever agronomic relevant scales, right? So breeders, we work with breeders to see if these tools can be used in a breeding pipeline. We work with the Iowa Soybean Association and Iowa Corn to see if farmers can be early adopters of some of these tools. And then that goes to the adoption part. We have social scientists who are involved in understanding what are the barriers to adoption and what are the catalysts to adoption of these AI tools. And finally, we think a lot about, and this is something that will come across in a couple of my slides, how do you democratize these tools, right? So training large models takes significant amount of research, significant amount of resources. So once you train it, can you enable a broader scientific community to leverage the training resources, right? And so the first example, I'm going to split the talk in 2 parts. The first part is about talking about what is the value of data. In particular, what is the value of unlabeled data, right? So as this community probably knows, annotating data is probably the major bottleneck to pushing our science agenda, right? So we can collect a lot of data now, but annotating the data, especially if it requires a PhD level skill set, is probably the most pain-consuming part of the research enterprise. So here I'm going to illustrate how perhaps large corpus of unlabeled data collected in whichever way we can think of, and in this case it's citizen science, can actually be used to train foundation models that can then be fine-tuned to be exceedingly accurate with very, very small amount of annotated data. And the use case here is something that is led by Aarti Singh, is can we come up with a comprehensive, robust platform for identification of insects, and we have now extended this to weeds also. So why is this a critical question? That's because accurate classification and detection of agriculturally relevant insect species is super important because there's increasing prevalence of overwintering, and especially with increased trade, there is the chance of potentially invasive species coming into a specific agricultural ecosystem. And so this is a tough problem, right? So when we started thinking about this, we identified 8 or 9 challenges, right? Why is this a tough problem? First one is, you see the images on the top row there are basically exactly the same species of bean leaf beetle, but it's expressing different morphologies, right? So you see variations in color, variations in size, variations in shape. And so having a simple vision-based, rule-based approach is not going to work here. Similarly, on the other hand, you have a lot of interspecies similarity. The second row, essentially, 2 different kinds of insects that share a lot of species, right? That share a lot of features. Other thing is the life cycle, right? So the insects can exhibit different life stages. They can be nymphs, adults, or here they can be larvae, and then they can be butterfly in this case, right, or moth stage. And then the last one, at least in this slide, is what happens when you have multiple overlapping insects, right? And here, it's not necessarily just identification, but once you reach an action threshold is when you want to take mitigation action, right? So you need to be able to count the number of insects, and overlapping insects make it difficult to do accurate counting. And so there are several more challenges here, right? So, and these challenges on the right are more to do with the data science challenge, right? So there are a large number of classes. If you look at reasonable count of the number of insect species, easily runs into the hundreds of thousands of species, right? And if you identify the agronomically relevant species, at least in the Midwest, right? So that's about 2,500 species. And so the other thing is collecting data. If you think of a butterfly, people like to collect pictures of a butterfly. But if you think of a bedbug, no one wants to collect pictures of a bedbug, right? So there's a lot of what we call class imbalance. Some charismatic species have a lot of data. Some non-charismatic species have very, very less amount of data. So how do you deal with something like that, right? And so the hypothesis that we essentially worked on is, can vision models that are large vision models, that is multiple millions of parameters in them, can we pre-train them using unlabeled data? So this could be just images, raw images without any labels, or the labels can be noisy, right? And then can you build this foundation model that can be subsequently be fine-tuned with very, very limited amount of data? And so where do we go for this data? So this is, as we were looking for, so Aarti's crew was collecting a lot of data within Iowa. We identified this data set called the iNaturalist data set. If you're not familiar with it, please go take a look at it. It's an amazing data set where citizen scientists collect images and then they post it up, and at least a couple of reasonable experts evaluate whether it's a good image, and then they annotate that image as and when possible. So we have about 230 million images available as of last count, as of May, June on INAT. Of that, you can get the the insects belonging to the Insecta family. That's about 13 million images. And Arti then identified about 2,500 insect species that were agronomically relevant. That is now 6 million images with noisy labels. Right? So, so what is the basic idea of self-supervised learning here? So you want to train a machine learning model. Usually when we think about supervised learning, you have an image and you have a class label. Right? And you train a machine learning model that says, given these images, can it make predictions on the class? Right? However, self-supervised learning, that's the top part of the right-hand side figure, essentially says, instead of coming up with a label, you come up with a proxy task. Right? So that forces the machine learning model to understand certain features of the image without actually having a label. What's a simple proxy task? Right? So the earliest proxy task was, I have a picture of a bird. I rotate the bird by 90 degrees. I force the machine learning model to say, how much have I rotated it by? So I don't need a label for the bird. Right? So then it understands what the features of the bird are and understands what the rotation means. And so basically the machine learning model is forced to learn key features of the images set that it is being trained on. So this is at least my key numbers slide here. So what is the value? Right? So we wanted to figure out what is the value proposition of a lot of unlabeled data, right? Or with either no labels or with noisy labels. And so we started off increasing the amount of data that we used, unlabeled data that we used to train a machine learning model by factors of 10. 60,000, 600,000, 6 million, and then currently we're doing 60 million and 120 million. Right? And so the results are, and then we used that pre-trained model and then fine-tuned it on 600K images that have been labeled very, very carefully. And so you see in the second column there, the clear value proposition of how the accuracy is improving. Right? The amount of labeled data is exactly the same. The amount of unlabeled data that is pre-training that you're using to pre-train the model is increasing, and you see a significant improvement in accuracy, right? From 89% accuracy to about 96% accuracy. Okay? So to us, this was a very clear empirical evidence that more unlabeled data is actually quite good. And this, and you can collect a lot of unlabeled data. So people, if they're not willing to share genotypic details, share management details, but they can still share just the raw data, and that's going to be pretty useful. So how does, then after we fine-tuned our model, remember this is over 2,500 classes, the model is able to clearly distinguish between 2,500 different insect species, and the figure on the top right is what is the accuracy per class. So for every class, we took about 10 images. We evaluated how well the model is able to identify which class it belongs to. And you see, overwhelmingly, for more than 90% of the insect species, you get more than 90% accuracy. And so what we also found was two interesting things. First thing that we found out was this is independent of the size of the insect. So the figure on the bottom is showing you essentially the violin plot, the accuracy plots of, we categorize the insect classes into various sizes, right? So, and this is a beautiful result because when citizen scientists upload images, they are either close to the insect or far away from the insect. So the data set itself accounts for various sizes and poses and so on, right? So, so the last point in this slide is, what is the computational effort needed here, right? So it took us significant computing effort, about 20 GPUs. These are 800 GPUs, 80 gig GPUs over 35 days of computing. But once you have the foundation model, training it is super simple. Less than half a day to train for your problem. And so it's able to identify several invasive species as indicated by USDA. It's also able to look at significant intra-class dissimilarity. All the figures in the circle there are the same beetle, and it's able to identify that with more than 98% accuracy. And then it's also able to identify species exhibiting similar features, right? So the 3 rows there are 2 different insect species that are extremely similar. So remember the fourth theme that I said was about democratizing these tools. So we developed this foundation model. We have open sourced all the weights, and we have also made sure that the models that we have trained is put between a front end that's easy for end users to use. So you can just upload the image. It's going to tell you what the class ID of that insect is going to be. So we last year or 2 years ago, when we got a reasonable version of this working, Aarti took it out and showed it to a couple of farmers. So the first thing that a farmer essentially did was they took out their earbuds and then took a picture and said, "What insect is it?" Right? And immediately it said, "This is some insect." Right? And that's pretty damaging, right? So if you want the end user to actually trust this tool, the first thing that they're going to see is kick the tires and try to see if it actually works. And so that's when we decided to have guardrails on these tools, right? So especially if you're pushing it to the last mile. And so how do we put in these guardrails? Through several wrappers. The first is what we call uncertainty quantification, or essentially if you have a new image come in, if it does not look like it's in the distribution of the images, the large set of images that we have trained on, we gracefully say we're not going to make a prediction. And that's basically what we do, is called out of distribution detection, okay, or detection. So essentially we understand what the features of all the insects look like. If a new image comes in that does not exhibit these features, we gracefully, and this is a quantified way to something called the energy and entropy, and through that we refrain from making a prediction. Other one you see on the right is what we call conformal prediction. Sometimes the certainty of prediction is very low, and so the way we circumvent that is we say, instead of making a prediction of one single class, can I make a prediction that it could be class A or class B or class C? And together, I can guarantee that it hits some certainty. That's what we call conformal prediction, and that's what you see there, that it's unable to make a prediction on a single class with certainty, but with a 98% certainty, it's able to make a prediction that it belongs to one of these three classes. Then the next step that we did was, sometimes you don't need to be able to identify 2,500 insects. Sometimes you only need to identify which of these 65 insects are most important for my breeding program or for production, right? And so often we're, our growers, at least within Iowa, we're interested in a subset of insects that are in the field crop insects. So we took this global model and we fine-tuned the global model with very, very small amount of data. Right, so we fine-tuned it with maybe 50 images per insect or 100 images per insect. This is what is called K-shot training. Right, so you only need K for a small K number of shots of images of a certain class, and then we showed that this actually beats state of the art significantly. Again, the value proposition of unlabeled data, training on large amounts of unlabeled data, followed by small amounts of fine-tuning. We used a museum data set, which has about 291 classes, and you can see the first column there, a significant improvement of nearly 40% in terms of accuracy. Then the question came in, okay, I'm able to identify an insect species, so what? Right, what do I do with it? Right, from a grower perspective. That's when we thought, could we integrate that with a conversational agent, with large language models, right? So you have a vision model that takes a picture, comes up with a class label, class ID. We can then put this into a chatbot like ChatGPT, and that's what you see here. The picture on the left is our pest ID framework, where you upload an image, it tells you what the class ID is, and then it gives you some information about that, and then you can start conversing with it. So the problem with current conversational agents is that they tend to hallucinate, right? They give you a lot of unreliable information. So how do you ensure that the information that's being provided is actually reliable? And that's basically, we use something called retrieval augmented generation. That is, again, Aarti and Danny's crew went out and collected a lot of extension data sets, right? That is pest mitigation strategies for individual insects. And so once we have identified which insect it is through the vision model, we provide the document into the ChatGPT and say, answer any question that the end user asks, but within the context of what is in this document. Right? This is what is called retrieval augmented generation. And so what you see on the picture here essentially is it's identifies the fall armyworm, and it says, is this a pest in Iowa? It gives a response, but it also says where it has taken the response from. So especially if you are giving some mitigation actions, use this kind of chemical, you can actually go, the end user can actually go and check to see if that is the most relevant piece of information, right? And so we evaluated this on multiple large language models, both on open source like Llama, as well as on closed source like GPT and Claude. And we found out that Claude seems to be working pretty well with this. And next is what's next after this, right? So we have essentially a large foundation model, but then you want to deploy it on the field, say on autonomous platforms, right? And when you want to deploy this on autonomous platforms, the memory on these edge devices is very small, right? So what we do is we perform what is called quantizing, right? So we reduce the size of the neural network so that it fits into edge device. And what you see on the left is a simple multi-arm articulated robot that's actually with a camera. That's
to develop new and resilient and
sorry, I don't need the video, the voice there. Thanks. So we are able to estimate how many number of insects there are per unit area to provide mitigating information. Full circle. So we started off with a couple of hundreds of thousands of images. We went to 13 million images. And so seeing the results that we got, we decided to go full throttle on collecting as large a data set as possible. So we are curating most of INAT with what is called coupled language image labels, right? So we have, for each image, we have both the Latin name as well as the taxonomic tree information as well as the common name, so that we are trying to collect about 135 million images. That is called BioTrove. So it's available for any of you all to use. It's in a AI-ready format, download and ready to start training. So this is something that we have packaged. So the key outcomes of this past couple of years' effort is large data sets. Again, the value proposition that I want to emphasize is, as a community, we're collecting a lot of data. We don't have to share all the metadata. Just sharing the raw data itself is going to be super useful. And then we have identified several approaches for guardrailing our predictions in terms of uncertainty quantification, conformal predictions, and ensembling. And then we have democratized this through a Pest ID app. So you are free. Please, I welcome all of you to try out the Pest ID app and give us feedback. Right? So this can be used also. It does 2,500 species, so you can also try it out in your gardens. Right? So what I talked about, that's my first part of my talk, was in terms of phone-based, handheld-based, quote-unquote, phenotyping. Right? So now I want to go in the 12 minutes that I have remaining one higher scale. Can we build using the ground-based imaging, drone-based imaging? I have one slide on our work on satellite-based imaging. So here, the two broad themes I wanted to talk about was this notion of foundation models. The second is the utility of 3D geometry, 3D representation, right? And so this is, as a mechanical engineer, I'm a big fan of 3D structures, right? But there's a very, very rich history of mechanistic models in agriculture, right? Going back hundreds of years. And so there are several applications that can utilize and leverage these 3D models. A partial list is there, right? If I have an actual 3D representation of my plant, I can do better light use efficiency. With a good crop model, I can then do a much better photosynthesis use efficiency. I can figure out what is the interaction of my canopy with winds. Especially, this resonated a lot with me after the derecho that hit Iowa. And so that's something that I was very interested in looking at. How does the internal structure of, say, soybean and maize plants affect whether it's going to be large or not large, right? And then the other thing, and you'll hear perhaps some interesting talks later today on how do you fit this to sparse data, right? So if you have sparse data, LiDAR data, using these 3D models, you can actually get phenotypes even based on sparse data. So what are the challenges? So at least from AI and computational science perspective, I see 3 challenges. First is, how do you actually reconstruct the structure, right? We know how to take 2D images very well. We are getting better and better at using those 2D images and using LiDAR to construct 3D images. Second is, okay, we construct this 3D representation, but is it in a format suitable for analysis, right? So just point cloud data might not be necessarily useful for downstream analysis. And third is, well, you actually have to do fast, high-resolution analysis. So how do you do these things? So I'm going to quickly go through each of these challenges, our attempts to circumvent those challenges, and open questions for us as a community to think about these challenges. So there are several. First is, how do you reconstruct the 3D structure? So there are several approaches that the community has pushed forward. First is stereoscoping, right? So you take two, just like the eyes, you take 2 cameras that are looking at the same scene, and you try to reconstruct something in 3D. Next is structure for motion. So you take multiple images and you use something called col map. So you identify the poses, and then you reconstruct the 3D structure. Other one is multi-view voxelization. You voxelate the structure based on multiple views. And the last 2 are something that I've noticed that there are several talks, posters on these 2. First is the LiDAR, right? So and that's what you see at the bottom there. We had a very nice talk earlier in the day that explained the details of how LiDAR works, right? So essentially, you get a point cloud, a gappy point cloud data, right? And then newer approaches called neural radiance fields and Gaussian splatting. I'm not going to go into the details of those, but Wei Guo, Ian Stavnos both are here doing wonderful work on these 2. Please talk to them, right? And so, but from a affordable phenotyping perspective, from a frugal phenotyping perspective, LiDAR is probably a difficult one, right? So we purchased a LiDAR a couple of years ago for $70,000, right? So, and this is very, very useful. So all our ground truthing is based on that. But what you see using these newer approaches, neural radiance field and Gaussian splatting, a simple camera, a phone camera, will be able to get you there. So some examples here essentially are, so what are the challenges with this neural radiance field, right? So we have had some success. So the figure on the left is video around a plant, and then what you see in the middle is the LiDAR scan, which we call the ground truth, because we very painfully took LiDAR scans from multiple views to reconstruct this point cloud very, very carefully. And then the figure, the second one there is your nerve-based reconstruction, and we have come up with metrics, right? How do you evaluate how well your reconstruction is based on this ground truth, right? So we have notions of precision and notions of recall, and so these serve as a valuable benchmark for the community to start developing better techniques than what we have currently, right? So the challenges are, how do you reduce the training time from 8 hours, 6 hours to something smaller, right? How do you create a watertight mesh? And the other thing that's also talked about earlier on is we are dealing with slender objects, right? So leaves move, and so getting the structure of slender objects is a non-trivial problem, but there's also a lot of opportunity to engage with engineers who deal with slender structures. And so we tried out, again, we collected a lot of data that we are making public, and this is highly correlated data. So we have images, we have the poses at which we're taking these images, we have the 3D reconstruction from LiDAR, and we have done this for 1,000 individual plants, corn plants, with all these data sets that are, so look out for the data set that could be useful for any of the calibration and evaluation of techniques. So that's the challenge of reconstructing geometry. Once you reconstruct geometry, what do you do with it? How do you essentially come up with in a good representation of this geometry, right? And so the way we currently do this is either deal with the point cloud itself, or we construct what is called a mesh of the point cloud, right? And the mesh can often be noisy, and sometimes it's not watertight, there's gaps. And so how do you circumvent that? There's several ways of circumventing this. One nice way is what is called the sign distance field, which I will not talk about today. Other approach that we are taking is something called using, as an engineer, we use a lot of CAD models, computer-aided design models, right? So this is essentially what we call CAD models are simple graphs with knobs that can represent complex shapes. By changing, for instance, if I have a cup, a CAD model of a cup, I can change one knob, change the diameter of the cup. Change the other knob, change the size of the ear. Change one more knob, change the size of the height of the cup, right? So in exactly the same way, here one of my students created this procedural model of a corn plant, where instead of having 500,000 points to represent this image, I'm just using 500 points to represent this, so 500 knobs. I can change the knob, I can change the location of the leaf, the size of the leaf, the wiggliness of the leaf, the number of leaves, the orientation of the leaf, and so on. So this is, in my mind, a very elegant way that's low dimensional, right? So you're able to come up with a low dimensional representation of a complex object. It's explainable because this knob affects the leaf angle. This set of knobs affect phyllotaxy, right? So it's very explainable, and it's super computationally efficient, right? And so given this is recent work where given a set of point cloud data, this is 500,000 points from a LiDAR, we are actually able to come up with a fitted model, this procedural model, that is represented by only 500 points, 500 parameters. So just imagine the 1,000-fold reduction in memory, right? So for every plant that you can, instead of having a full 500,000 points XYZ representation, you just need to have 500 parameters and a simple model code that reconstructs this geometry. And so for examples, for those of you who are corn experts, right? So we tried this out for a couple of different genotypes, the B73 and MO17, and you can see the variations in architecture that's accurately captured. Now, the last challenge in the 3 minutes that I have is I talked about how do you reconstruct the shape, how do you represent the shape, and third is what analysis can you perform with the shape. Right? And I'm going to focus on the left side, where again there's a lot of work by a variety of groups across the world. One thing that who are reporting open source code. So I'm a big fan of open source. So this is Helios from Brian Bailey's lab in UC Davis, which is essentially a very fast biophysical modeling framework, especially radiation transfer. So the problem that we are looking at is, can I quantify PAR on a set of diverse varieties that have grown? This is work with Pat Schnabel's group. The way PAR, at least to my understanding, is measured is you have a meter, light meter above the canopy, you have a light meter below the canopy, you walk across the row, and you figure out the difference between the light intercepted at the top of above the canopy and light below the canopies, right? So we can do exactly the same thing with my virtual twin in this case, right? So I have a 3D representation, I can do this exactly the same thing. So you place the reconstructed plants on a virtual plot, and then mirror the actual arrangements of the plot, and then perform analysis. Note that when I'm doing the part, especially for large field trials, I'm only measuring empirically, experimentally, at one time point. Okay? But what this allows us to do is, or before that, maybe I show that we showed that the virtual part is correlated very well with the actual experimentally determined part for a diverse set of genotypes. Right? Then we can take this out for a ride. Right? We can say, I can place plants at different phyllotaxies and figure out what is the implication on PAR. I can also look at PAR over the whole day, rather than figuring out photoactive radiation at one point of time. Right? So I can figure out, and this is specific to location. This is all done for Ames. Right? So where I know exactly the lat long, the time of the year, that tells me what the motion of the sun is. I can figure out how much photoactive radiation is incident. So what kind of analysis can we do? Right? So we can explore the effect of phyllotaxy. Right? So changing the orientations of the leaves impact the amount of power that's being that's absorbed or that's incident on the plant. Similarly, this growing season, Pat's group utilized some of these analysis to figure out at what times of the day is it most ideal to perform these experiments, right? So we can inform field experiments. And then the last one that I'm very excited about is this procedural models. Remember, there's only 500 variables, super easy to perform optimization, so we can perform optimization, right? Can you, given a certain row spacing and seed spacing, and given a certain biomass constraint, can you design how the leaves can be so that it maximizes the light use efficiency? This could be useful information for breeders to start playing with, right? So is this actually an idiotype that I want to breed for, right? And this can be extended to a few other things, light use efficiency, to photosynthesis efficiency, to lodging, and so on. And so a couple of things, other things that I'll very briefly talk about. Getting these 3D reconstructions provide a wonderful opportunity for education, right? So the figure, you can put these 3D models into artificial AR, VR, right? Augmented reality, virtual reality modules. And you can look at various physiological stages. And this is, in this case, this is with Danny Singh and Adarsh Krishnamurthy, where they're looking at millets, staging millets. And you don't have to only wait for those 5 or 6 weeks in the year, at least in the Midwest, where you can actually stage these, right? So you can actually, any time of the year, winter, you can actually stage this and understand this. And so in the last minute or so that I have, the last problem that I want to talk about is we have undertaken a fairly intensive data collection campaign that the data set is now available, where we have taken across 6 locations in the Midwest with multiple nitrogen management conditions. At exactly the same point that we're collecting satellite data, we're also collecting drone data and ground data. So this is essentially data that is completely correlated. And so this allows us to figure out the value proposition of satellites to drones to ground-based measurements, right? So with that, I thank you for the opportunity to talk. And the 2 key points that I made was the value proposition of collecting raw data and training foundation models, and the value proposition of doing things in 3D. Right? So I'm happy to take any questions if there are.
Thank you so much, Baskar, for excellent talk. And we are running 5 minutes over time, so we will have plenty of time for Q&A during our keynote discussion, which will be at 4:35 p.m. today. So our next speaker is Aaron DeSalvio from Texas A&M. He is a PhD student in Dr. Seth Murray's lab. Aaron will present on temporal image sandwiches, enable link between functional data analysis and deep learning for single plant cotton senescence. Please welcome Aaron to the stage. And he's already at the stage, and please welcome him. Yeah, thank you.
All right, thank you for the introduction. I'm here with my advisor, Dr. Seth Murray from Texas A&M University, and I promise I will explain the title later in the presentation. So senescence is a term that describes the orderly deterioration of biological function, and in a crop like cotton, it's particularly important for fiber quality and yield, and in cereal crops, it's arguably even more important for grain quality. The identification of a fast senescing genotype in cotton could have relevance for the organic sector in particular, where defoliant usage is discouraged or not allowed. And, identifying one with divergent temporal phenotypes could also enable us to ask more basic biological questions, such as the molecular genetic basis of senescence differences between genotypes. So like many breeding traits, as everyone in the room knows, it's a very laborious trait to collect, particularly due to the revisit time that's required to obtain senescence data. And so the advantage of using a drone or a greenhouse is that we can repeatedly score senescence after the fact using both visual scores or vegetation indices. And so this picture is meant to convey the throughput with which you can collect senescence. So each individual square here is a single cotton plant. And you can track the senescence across time, moving from left to right, according to days after planting. So this project takes data from 2 different experiments that were conducted in College Station in April to September of 2023, and I'll refer to them as E1 for experiment 1 and E2 for experiment 2 moving forward. So experiment 1 was a panel of 48 genotypes, all of which had a Gossypium hirsutum background. These are BC5 advanced backcross inbred lines. They all contained small portions of the wild species, Gossypium tomentosum. And this was a mixture of chromosome substitution lines, or CSLs, chromosome segment substitution lines, and then 2 pure elephant lines, as well as a Texas marker 1 check. And these were 48 single-space plants. There were 5 blocks, so this was an RCBD with 240 single-space plants. And then the outer rows and end hills were just there for experimental filler to absorb environmental variation. And so the great thing about working with CSLs and CSSLs is that we're operating under a common genetic background, so the Gossypium hirsutum background, and we can pinpoint fine regions of introgression. So in theory, the phenotypic variation should be due to the introgress segments that are coming from the wild species. E2 was very similar, just consisted of a smaller panel, 27 genotypes replicated 9 times. These are, again, advanced backcross inbred lines, again with a Gossypium hirsutum background. But this time, the introgression segments came from Gossypium mustellinum. So we followed a standard phenotyping pipeline using an RGB drone, and we were able to get 14 late-season flights that we used to stitch those images into orthomosaics that were used for senescence processing downstream. So with those orthomosaics at each of the 14 time points, we created single plant shapefiles, and that allowed us to track senescence for each individual plant within each genotype at each of those time points. And so to first collect senescence data, I implemented a very simple visual scoring system, which would be typical with a breeding trait. So this was a 0 to 5 scale, so 6 different categories. And combining E1 and E2, that left us with 473 plants that survived until the end of the season. And multiplying that by 14 flights, we were left with an initial data set size of 6,622 visual senescence ratings. And I'll abbreviate those as VSRs moving forward. So while I was scoring senescence, as we've mentioned, the annotation is the most painful part. I did start to take notice of divergent temporal phenotypes. So if you can look in panel A here, the plants senesced to an irreversible state of senescence. They didn't make any sort of recovery. But B and C represent the fact that cotton is a perennial, and so it can actually have indeterminate senescence at the end of the season, which is why defoliant is commonly applied. So in B, we can see that it senesced to about a 2 or a 1, and it maintained that until the end of the experiment, whereas in C, it actually senesced partially, but made a recovery toward the end of the season. So as many of you know, every time you capture an image, it's actually decomposed into its component bands. So in the case of an RGB image, we have red, green, and blue to work with. And so I looked at 34 different vegetation indices and assessed them for pairwise correlation with the visual senescence rating, because I wanted to try to find an empirical replacement for the visual score. So the 2 indices that emerged on top were the red chromatic coordinate index, and its formula is given up at the top right, and then the transformed normalized difference green and red index, which is TNDGR, which actually had the highest correlation, although it was negative in magnitude. So I chose to work with those moving forward. So the issue, regardless of the type of senescence metric we're working with, is that the data set is noisy and discontinuous. And so you can see that on the left, we have the visual score and then the 2 different vegetation indices. And regardless of which you're looking at, it's not a true representation of the underlying process. So we want to find a way to recover the smooth underlying phenotype that actually makes up senescence, because it's not happening in a discontinuous way. And so the tool that we're going to use for that is functional principal component analysis. And here's the formula for that. So this is, in theory, we're decomposing a function into a set of basically the mean function plus a set of deviations from the mean. And so it's, in practice, we're going to keep a very limited number of principal components, so functional principal components in this case, and that's indicated by K. So we've gone from an infinite sum, and we're retaining just K functional principal components. And so again, this is the mean function for each plant. So the ith curve would just be the individual cotton plant in this case. And we're representing that as the mean. So that's the average temporal senescence trajectory. And then the functional principal component score for each individual plant. And then the eigenfunctions are the overall deviations from the mean. And then the scores weight each function for each plant. So it tells it how much of a deviation is occurring from the mean. So one of the great things about FPCA is that while we're recovering the underlying temporal phenotype, we can also reduce the dimensionality significantly of our function. So this is just what the raw data looks like for an individual plant. So regardless of the metric, we have 14 scores for each time point, for each plant rather. And then after we perform FPCA, in this case, we're going to retain the first and second functional principal component scores. And again, these are the Greek letters xi in the expansion, and those are just telling it how much to deviate from the mean function for that specific plant. And if you'll notice, FPC1, regardless of the metric, is a few orders of magnitude greater than FPC2. And the reason for that is it explains the vast majority of the temporal variation. So if you look, FPC1 for the visual score is explaining 90.2% of the variation, and then it moves down to 83.5% and then 87% for TNDGR. So regardless of the metric that we're using, we can essentially categorize the senescence trajectories into temporal categories by basically sliding along the x-axis on FPC1. We can obtain plants that are undergoing a stay-green phenotype or senescing rapidly. And so I've denoted that with an open or a closed circle. And you can see that RCC is a lot messier than the visual senescence rating, but TNDGR does a much better job of clustering. And in the case of TNDGR, recall it was a negative correlation, so that's why the open circles are over on the left side. So we were able to recover the smooth phenotypes for senescence for each individual plant. So this is just all of the plants in the experiment shown on the left here. And then I pulled out a few specific plants and colored them by genotype to show that there may be a sort of genotypic component that's regulating senescence. And of course, in the rapid senescence case, there were a few outliers, but 4 out of the 5 replicates in panel B show that there's a genotypic component to the senescence trajectory. So the next question I wanted to ask is, can a deep learning model learn the same temporal patterns just using the images themselves? But the issue is that we have a dimensionality mismatch. So we're treating the first functional principal component score as a reasonable summary of senescence, again, because it explained between about 84 and 90% of senescence, but each plant still has 14 different images associated with it. So we have a mismatch there. And so what we're going to do is, again, we're going to use the array structure of an image to our advantage, and we'll separate each image into its component bands, and then we're going to stack them according to dates. So basically we're taking each image at each time point and we're concatenating it along the third axis. And so we basically have an array that goes from the XY dimensions times 3 to 3 times 14, which is 42. So we have an X, Y, Z, and our Z axis has now accommodated the temporal aspect of the images. So we're representing them as a time series image. So this is the recipe to make the image sandwiches that I promised you. And again, the dimensionality of the image set is compatible with the FPC1 scores because every single plant has a single time series image to match that FPC1 score. And so I was very aware that the data set size is not in the millions for this experiment, so I underwent some data augmentation and essentially boosted the data set size to around 2,300 time series images. And I'd like to emphasize that although we've reformatted the input size, we haven't lost any of the spectral data because we've simply increased the depth of each image, but we haven't removed any of the time points. So before I trained this model, I got very lucky and ran into a paper that discussed hyperparameter optimization. And so I found a Python library that specifically discusses this called Optuna. And essentially you can take a subset of your data and give it a set of hyperparameters to search through. And so I modeled it after that paper that I had read, but essentially you can give it these choices. They can either be categorical or continuous distributions for something like dropout or learning rate. And you can empirically determine the hyperparameters that are going to work best for your model instead of doing it ad hoc or just using the defaults. So again, I wanted to continue to combat the modest data set size and take an aggressive train test split. And so I used the experiment 1 data set for determining the hyperparameters, and I used a 50-50 train test split with only those images. And then for the final model training, I asked it to output all of the E2 time series images using, again, those hyperparameters that we got from experiment 1. And so in the final model evaluation, I asked it to output all of E2 images, and then I did an 80-20 train test split with a 20% internal validation for experiment 1. So in total, I looked at 6 different models, and M1 through M3 are assigned to the visual senescence rating, RCC, and TNDGR, and then M4 through 6 are just copies of that, but the difference is that I allowed the dense layer to be searchable by that hyperparameter optimization framework. So the models are very similar, but I was just curious if the specification of the dense layer would affect the outcome. So another great thing about the hyperparameter optimization is that it doesn't just give you the parameters, but you can actually get the variable importance of each parameter. And so it's kind of looking into the black box in a way. And so as we expected, the learning rate had the most important outcome for visual senescence rating and RCC, but interestingly, that was not the case for the transformed normalized green and red index, where other features such as the number of layers and the activation function had an influence on the outcome. And so to me, it was worth it then to do the optimization because there's no one-size-fits-all set of hyperparameters. Okay, and looking at the metrics for each model was evaluated 25 times with a randomized train test split. Based on R-squared alone, we can see that the visual senescence rating performed the highest with a very low mean absolute percentage error. M1 performed very similarly. In second place, the TNDGR index did pretty well with a 0.74 R-squared, also just below 6% for the mean absolute percentage error. And RCC did not do as well. And that didn't surprise me again, because if we look back at the FPC1 versus FPC2 plot, we could see that RCC was not able to separate those temporal phenotypes as well as the other index was. So you can actually treat the functional principal component scores as a data set. So a lot of times people will do multivariate, or in this case, univariate analysis. So I kept the FPC1 score, and I actually looked at analysis of variance for both the actual FPC1 score and then the predicted score from the deep learning model. And we can see that there is, in fact, a genotypic component to the temporal senescence trajectory, at least in this panel of CSLs and CSSLs for cotton. And interestingly, the repeatability was actually higher after using the predicted values from the deep learning model. So I generated saliency maps and this is indicative of all of the images in the stack. And we can see that, at least for the top performing model, the model is looking exactly where a human scorer would look. So it's encouraging to see that throughout the temporal domains that we added, the model is looking at the appropriate pixels for making its predictions. So again, to wrap up, we were able to use FPCA to significantly reduce the dimensionality of our time series data set while simultaneously recovering the underlying phenotype. We found candidates for replacement of visual senescence ratings with those two indices, although TNDGR did substantially better. The regression models were able to, in most cases, map back the time series image stack to the FPC1 score, which was a summary of our temporal senescence phenotype. And then looking at analysis of variance, we confirmed that there was a genetic influence on senescence trajectories. And I would argue that FPCA can serve as either a basic or an applied tool, because you can actually look at the temporal phenotype as opposed to evaluating phenotypes at static time points repeatedly. So there were definitely some limitations to the approaches that I used. For example, it was pretty computationally intensive to run a lot of Optuna trials because I set so many different parameters to search through, so we may not have converged on the optimal parameters. There was, of course, noise inherent in the drone images, and that could have reduced the regression model's ability to map back the pixels to the FPC1 score. And this was just these were simple convolutional layers, and so perhaps using LSTM layers or gated recurrent unit could help with more complex models. And in addition, this data set didn't have any sparsity in the sense that every plant at those 14 time points had an image, but it would be good to test this by removing a certain percentage of images at each time point to test its robustness to missing data. A lot of people went into making this work possible. I'd like to thank my advisors here today. I'd certainly like to thank the Stelly Lab for letting me use their germplasm, and I'm very grateful to all of you for paying attention. So thank you for your time.
Thank you so much, Aaron, for presenting your excellent research. Questions for Aaron?
Hi, I have a question about the image sandwiches. So from your slide, I understood that you're stacking these 2D images on top of each other across the different time points. Do you think that maybe perhaps the order how you're stacking the different channels has some kind of an effect on the result or the modeling? I mean, maybe putting all the red channels next to each other or something like that. What do you think about that?
Yeah, it's a great point. I guess the default loading in at least in TensorFlow has red, green, and blue. So it would be very interesting to see if blue, green, red actually makes a difference. I honestly don't know, but it is a good idea to test that. Thank you.
Hi, here. So from what I understood, first, it was a great talk. You did the sandwiches by stacking RGB and then the time points only in the channels. Have you tried using 3D tensor? You know, you have channels, X, Y, and Z, and then stacking on Z. So you do 3D convolution instead of 2D?
No, that is a good idea, though. I appreciate the suggestion. It was just 2D convolutional layers, so
yeah, in that way you can use the channels as they are, and you use the same idea, but you have the Z axis to do the time steps, and then you can do the regression. So the channel order won't matter.
Okay, thank you. I appreciate that. Sure.
Over here. So what network architecture did you use? You mentioned some other ones to use, but what convolutional based architecture did you use? Like recurrent neural networks, or
it was strictly convolutional. I didn't actually use LSTM or GRU or anything like that. It was strictly a convolutional net.
Did you use a ResNet architecture? There's lots of different architectures that you can use that can provide different benefits. Did you explore different architectures? The GRU and those are very, very different than convolutional based.
No, I understand. I did not pre-train the model. If that's what you're asking, it was strictly trained from scratch.
Okay, thank you.
Hi, great presentation. Did you account for background in the images? Like, I don't know if the soil would be a factor in this, or did you binarize it such that you're only analyzing the plants, not the background?
Yeah, it definitely would affect it. I had issues with I had issues with masking out the soil because the senescence phenotype almost looks identical to the soil. So I would like to go back and redo everything with masked images, but that there definitely were contaminating soil pixels in there. And the activation maps show that in some of the other models.
Aaron? So I have one question, Aaron. So how you made sure that senescence is just because of nature or naturally, not because of any disease? Because the crop I work, we see this kind of senescence a lot when we have these fungal diseases, leaf blight and leaf spots. So.
Sure. Well, I visited the field to confirm that it was senescence, but it's certainly possible that I missed out on a disease in that particular field. It's known to suffer from blights and things like that, but I'm pretty confident that it was natural causes. Yeah.
Excellent.
Yeah, okay. Sure. Here. So used RGB, but I think if you would use an infrared channel next to the RGB, you would get an easier soil plant segmentation. I think if you would put the NDVI only in your training network, it would be more straightforward. Now you're not sure what your model is actually using as a determination of the senescence level.
Yeah, I agree with that, and it was just due to I kind of improvised that experiment last minute on somebody else's data set, so I did fly this summer using a multispectral drone, so yeah, I'll incorporate NDVI this time around. Thank you.
So if we have no more question for Aaron, let's thank Aaron for giving excellent talk. Thank you. Next, we have Dr. Elizabeth Sklar, who is professor in agri-robotics and research director at Lincoln Institute of Agri-Food Technology at University of Lincoln, UK. Dr. Sklar will present today on intelligent decision support for plant breeding using computational argumentation. Please join me in welcoming Dr. Sklar.
Hopefully you've got my slides. Clicker. Dreamland. Okay, great. Thank you. Now that I've got the technology together, thank you for having me. So I wanted to start by giving you a little bit of a geography lesson. So I am from the University of Lincoln, but not this Lincoln. And I am, do have an American accent because I have a dual passport, but I'm based in England at the University of Lincoln. And one of the things that when I was telling my colleagues that I was coming out here to do this, they were like, oh, Lincoln, Nebraska, that's the place that always comes up first when you Google University of Lincoln. So I just wanted to point out a couple of things that even though the University of Nebraska in Lincoln was chartered before us in 1869, we have a cathedral that was consecrated in 1092. So we have a little bit more history. But I'm based at a research unit within the university, which is called the Lincoln Institute for Agri-Food Technology, or LIAT. And we're based on a 400-acre farm where we have a lot of different kinds of things. So it's been interesting for me to see what's going on in this community. I have a background in AI and robotics. So I'm not a plant person, but we have our research institute is interdisciplinary. So we have a group of sort of AI robotics people. We have a group of plant science, crop science, weed science, agronomy people, and we have a group of social scientists. And our overall mission is looking at finding ways to sort of rethink agriculture using technology to help promote sustainability and forward agriculture for the next generations. And so what we realize is in order to have the, you have the agri side and the tech side, but you really need the social science side to look at the policy and economics in order to make sure that the solutions that you come up with in the research labs are going to actually be practical for real-time, real-world deployment. So we do a lot of different things. We have, I think, a pretty broad cross-section of research activity, given our size in terms of the number of staff members. And we do welcome visitors, so anybody who wants to come and spend some time with us in the UK, please come find me later this week, or you can follow up with email. So today I'm going to talk about some of the research that we're doing on a particular project which we call Miscanthus AI. It's focused on using, sort of bringing together some in-field imaging, some genotype work, and looking at how do you make decisions, how do plant breeders make decisions about the ways to increase the biomass for Miscanthus, which is an energy crop. So I'll talk a little bit about it. Now, I wanted to start by saying, as I mentioned, I'm the AI roboticist person on this team. And so I tend to focus on the things that are on the left-hand side of this chart. So look at data collection, look at data analysis, and then sort of hand everything over to the plant people and say, okay, you guys are the plant people. You can make the decisions and decide what actions you're going to take, and I'm not going to worry about it. So I'm going to talk a little bit about the left-hand side just to give you an idea of some of the data that we are collecting and the kinds of things that we're doing. But then actually the thing I'm going to focus on, the novelty, I think, is the plant breeder decisions and the way that we're bringing some kinds of elements of AI into that column. I'm just too short for this microphone, so sorry if I'm sort of out of breath on my toes here. Okay, so the crop that we're using is miscanthus. It is, so you probably all know more about this plant than I do because you're the plant people, but it originated in Southeast Asia. These are some plots that we're growing some in Lincoln, which is on the east side of the UK, and our primary collaborator on this project and the lead investigator is at University of Aberystwyth, John Doonan. Some of you may know John. And so some of our images come from Lincoln, and some of our images come from some sites they're running, they're growing in Aberystwyth. These are the Lincolnshire sites. And it shows the images that we took from UAV over a number of periods of time. If you're familiar with this crop, you know that it will grows very fast and it will senesce fairly quickly. It's a hardy crop, and it's because this project is funded by a program that's focused on AI for net zero. We're looking at this this crop that has high biomass. So we're doing in-field data collection. My group is looking at using UAVs and UGVs, and these are some of the equipment that we're using. So we have a DJI drone for doing the UAV stuff. It's got a MikaSense Red Edge multi-spectra camera, and it also has LiDAR on it. And then we have ground-based, which we have a robot that we have on we have an arm, cameras on the arm with the robot driving around in the field. And we've experimented with taking data with RealSense and with stereo cameras to see where we get some better data. So some of the tricks that we're looking at with the UAV processing is looking at, okay, so we'll get sort of this point cloud data, and what we really want to know with the miscanthus is we want to be able to estimate the height, so we can estimate the density of the plant and the volume for biomass. So we really want to be able to look at the height, and so one of the things that we had to do early on was look at, you can't actually just take the raw height from the UAV, because you have to take into account the height of the actual ground, so the terrain. So if the plot is growing a bit on a hill, you have to make sure that you're subtracting out where the terrain dips down. So that's what we're doing. That's corrected for the terrain on the, I do this? No, I don't know how to do that. Anyways, I'm not very competent with this device. Okay, so anyway, so that's the UAV data. Then the UGV data I mentioned, so we've got this robot arm, and this is a very small sample crop that we have growing in the garden part of our farm, where we can do some very controlled experiments and look at things very closely. So normally, as you can tell from like these plots, the miscanthus grows very closely together. And so if you want to have a ground-based robot actually take images of it, you can really only get around on the edge of it. You can't, they're too closely packed in, so you can't get them too close together. So the nice thing about our little experimental plot here is we're just growing one row, so you can get around and we can do some experiments. And what we're trying to do is build some 3D reconstruction of the plant. So with the sort of long-term aim of being able to have a 3D model so that the agronomists can just need to go out to the field as often as they do and get some of the sense. Of course, it's still fairly crude, but that's what we're doing at the moment. And then the main trick is to basically put together the UAV and the UGV data so that we can use the data that we get around the edge of the field from the UGV, pair that with the UAV data, and then use that as a way of interpolating what might be going on at the ground level within the inner part of the field where you can't get the robot inside. So this is some of the things that we're doing. one of the things, maybe it's just where's the button for there we go, yay, okay. So you can sort of see here, one of my postdocs came up with this. Basically, it's a 3D quadrat. It's a little bit hard to see, but it's got some red balls on it of different heights. So we're using those as key points to be able to match frames from the UAV and the UGV and use that to build this 3D model that you can zoom around in, which is sort of a very first cut there. So they'd probably be annoyed with me if my postdocs knew that I was showing this here, because it's just really hot off the press. But I wanted to give you an idea of some of the stuff that we're doing with this. Okay. And then once we've collected all the data and processed it, you do sort of standard data mining, machine learning kinds of things, looking at, detecting, classification, segmentation. Estimation is the key thing that we're doing for this project, because we want to estimate the volume of the biomass, as I mentioned. So none of this is new. It's just sort of standard stuff that we're doing. And here's a little bit of the examples. So we're doing this both with the UGV and the UAV data. The UAV data is interesting because this is the Aberystwyth field. This is where we get the data from more genotypes. And the stuff in Lincoln is a smaller set of genotypes. So we'll be able to look at some of the variation from the different genotypes, which we feed back to the plant breeders. So now I want to get to what's hopefully the more interesting part of the talk, which is this third column that I mentioned here. So the plant breeder decisions. Okay, and a lot of the research that I've done in my career is looking at AI decision making, which is actually not machine learning. So I'm going to talk about this. So we started by looking at what are the things that plant breeders might say. This is the example from Miscanthus. And so they might say different things about, well, it's more important to focus on yield than on disease resistance, and other people might say it's more important to focus on disease resistance as opposed to yield, depending on what your environment is, what your priorities are. So we interviewed a lot of plant breeders, and we're still interviewing plant breeders. So anybody who wants to provide input to our project, again, feel free to follow up with me, because we'd be happy to have a conversation with you. So we sort of took all this information from plant breeders, and they were like, okay, how can we help this feed this into our AI decision engine? And we look at, well, what are the things that AI people say? And so there are people who are sort of above a certain age, like me. When we started studying about AI, machine learning was a very tiny piece of AI, and AI has is much bigger than just machine learning, much bigger than deep learning. There's a whole areas of AI around planning and reasoning and problem solving, knowledge representation, and a lot of other aspects of AI. So I kind of jump up and down. I won't rant about that here, but I just wanted to point this out because I wanted to show you that some of the things that we're pulling together about reasoning under uncertainty, planning and reasoning, these all feed into decision making. So we look at how can you use AI for making decisions. So AI is good at doing things like making statistical predictions, but AI isn't perfect. AI doesn't have common sense. And AI is only as good as the structure, the data that feeds it, and the methods that train it. So you have to be really careful in terms of how you use AI and how you use the output coming from AI. And note that, not all AI is data-driven. So I think a lot of what I've been seeing here is very neat applications of deep learning and data-driven AI, but there's some other sides to it. And I could give a whole seminar on that, which I won't. But these are some of the areas of AI that you can use for sustainable agriculture, which is the sort of wider part of what we're doing on our research center. So we look at, monitoring crops, modeling crops, adapting behavior to how things change, and problem solving. So this kind of decision making in terms of how do you improve things because you want to increase yield, or do you improve things because you're more worried about disease resistance? And of course, they're going to go hand in hand to some extent anyways. But these are some of the kinds of things that we're bringing together. Now, the key technology that we're using for our decision making is a formalism called computational argumentation. Computational argumentation has its roots in philosophy and social science. It's originally a model of how people reason, and I do spend time going to conferences on computational argumentation, which is very theoretical. There are not many people in the world that are actually implementing computational argumentation for real-world applications, and not many are doing that within the agriculture domain. So we're one of the few groups doing that. But here's a very simple sort of coverage of what argumentation does for you. So what argumentation provides is a way of structuring evidence for and against certain conclusions. So you have the support for something and you have attack against something. It's often modeled as an argument graph, and it can be used to model sources of information, so maybe how much trust you have in a source of information. A while ago, we did some research looking at, well, it's not just enough to model the source of information, but you have to look at what is the context of the information that you're getting. So you might have a trusted source that could tell you really good things about soybeans, but they're going to tell you crap about sugar beet. So, then the source might be great for one context, but not for another context. And so some of the complexities that you can model within argumentation would let you do that kind of thing. But at an argumentation conference, you'd be looking at lots of things like this and like sort of formal statements like this, but much more complicated. And I know I'm standing between you and coffee, so I'm not going to bore you with that. But let's come back to the slide that I showed you earlier that said these are the things that plant scientists, plant breeders might say. And now I've structured it using an argument graph. So here I say, okay, so you have one group of people that say, well, they want to breed for disease resistance over yield, and an opposing group that wants to breed for yield over disease resistance. And these are some of the evidence, the reasons why you have yield over disease resistance, and these are some of the reasons why you have disease resistance over yield, okay? And you can see that there's not going to be a single right answer. This is one of the things I really like about argumentation, is because if there was a single right answer, then we just have a basic rule-based AI and it would be done. And to me, that's kind of boring. So I want to do something that is much more difficult and that has a lot of subtleties around it. And there's not going to be a single right answer depending on the farmer, depending on the context, the climate, the financial climate. The answer is going to be different. And so you want to be able to build a system that will let you deal with those kinds of real-world flexibilities. So we've built this little decision support tool. It's in a very prototype stage. And this is what our little interface looks like. So basically, you can select what crop you're using, and the knowledge representation that we've built is supposed to be sort of generic enough that you could use it for other crops. Because of our funding for this project, we're just looking at miscanthus, but the idea is you could select different crops. You could say, what are the kinds of things that you want to improve? So do you want to improve yield? Do you want to improve disease resistance? What's the location? Because again, depending on where you are in the world, your priorities may be different and the rules may be different because climate's different and terrain is different, et cetera. And then you can step through some different things that say, okay, well, if I have disease resistance being important, this is why. If I have yield being important, this is why. And so I can set my priorities here. I've got disease resistance over yield, but you can reset your priorities using a screen like this. So it just gives you a little bit of an idea. And this is designed to run on a tablet, and it's designed so that we would get input from the breeders as well as input from the fields to be able to feed into what's the right set of information to provide the breeder as you're doing this decision making. So just a quick summary. I think I'm doing okay for time. So we're doing some in-field data collection with UGVs and UAVs and trying to fuse some 3D models that will help breeders not feel they need to go to the field as often as they do now. We're well aware that a lot of times breeders need to touch the plant, especially with miscanthus. They want to see, how spongy it is and that kind of thing. So that's another project with, tactile sensing on soft robots, but I'm not going to talk about that today. But we always have to start somewhere. So these are some of the things that we're looking at. And then the thing that I think is probably novel for the community here is the application of computational argumentation to this kind of decision making where there's not one single clear answer. The key challenges with the argumentation part are knowledge acquisition and knowledge representation, because every plant breeder you talk to is going to tell you something a little bit different. And so how do we put that together? And that's where the idea of this argument graph is really helpful, because you can model the different inputs from different people and show where are things that are conflicting and where are the things that are supporting things that other people say. The other thing that's a challenge is that the knowledge represented is obviously highly specialized for each crop. So if you want to scale this for different crops, you need to change some aspects of the data set. Now, some things may stay the same, and some things, will be different. So, ask me next year when we've gotten more funding for a different crop. But right now, we're just looking at miscanthus with this. I'm going to close with a couple of shout outs for some projects that I'm involved with. So I know that there's been some presentations here from people representing different networks. In the UK, you may be familiar with Phenom UK, which is it's a plant phenotyping network, also connected with Emphasis. So we have a new one in the UK called AI Bio, or UK AI Bio, which has recently been funded. I'm chair of the steering group. It's led by Andy French in Nottingham and Ryer in Aberystwyth. And it's the key thing with AI Bio is that we're bringing in the AI parts, is not just plant phenotyping, which obviously is using a lot of AI, but there's also bringing AI people into the same network. So it's funded by UK government agency, but there is interest in international collaboration. So anybody who wants to know more about that, the website for that is here. And the final thing is we have a new doctoral training center, which is focused on AI for sustainable agriculture. So a number of people mentioned they're working with undergraduates. We are accepting applications for next year's entry, 2025, for a fully funded 4-year fellowships for PhD students. So if you have anybody who's interested, come talk to me. I'm a deputy director, and then here with a colleague who is the director of this center. So thank you. And I'll just, any questions? Just also want to acknowledge my postdocs in blue, who are the ones who've done actually the real work here, and the other colleagues on the Miscanthus AI grant. Thank you very much.
Okay. Thank you, Dr. Sklar, for sharing your cool research work. Questions for Dr. Sklar?
Thank you. Very interesting work. Just maybe adding a bit more to that breeder discussion to have maybe add to the confusing thoughts. So breeders, we often, we think in terms of an ideotype, right, which may not necessarily be readily found in the field in one cycle or one field season, but that's in the creative mindset. That's what we try to aim for, right? And it often doesn't come in the form of an ideal of a line, like I said, but it comes in the way of thinking of the identification of parents that have complementary traits that then build towards the formation of that ideotype. So I'm just wondering, in that, this development process, was there any discussion, or what's your insight in that regard?
So I'm not sure I completely understood what the question was.
Well, the question is, how does your the platform integrate the identification of traits that are complementary for for genotypes to be used as parents rather than the identification of progeny?
So that would have to do with how the data is fed into the system. And so we have the collaborators in Southampton are doing the work on the genotype data, and then we're linking that with the visual data that we get from the different cameras. And so the analysis works that's looking at what are the elements or the features of the images that you can correlate to the different genotypes. That's where we would look at, what's the derivative from one genotype from a parent to a child. Okay.
I have a question about how you address that this is the perennial grass and 3 first years miscanthus needs to establish, so number of the shoots will be different from year to year. And then somewhere around 10 years, 15 years, for some hybrids, the middle of the plant is empty and only on the edge the new shoots are coming. Are you looking for that? And also another question you didn't mention in this breeding, let's say the study connected with the breeding, on what species do you put your efforts? If they are the inter-species hybrids, or this is Miscanthus sinensis, what is the material on which you work?
So unfortunately, I'm the AI person, not the plant scientist, so I'm not sure I can answer. The project's been running for about 1.5 years, so we haven't hit the issue of, that you've mentioned about sort of 15 years down the road in terms of the how the plant senesces over time. But I'm happy to connect you offline with the plant scientists on our team who may be able to answer your question. Sorry.
Do you follow up a question? Do you focus on the traits like emerging, the time, or are you only focusing on the how many shoots you get, in the, let's say, middle of the season?
We have all of that data in the system, so it's just depending on, what are we're looking for correlations, and then to be able to let the plant breeders decide which correlations are helping them identify the kind of actions that they want to take.
So how early are you starting the phenotyping in the field? As early as the first shoots are coming, or you're waiting longer?
No, we start from emergence. We start taking images from, yeah.
Thank you.
Yeah.
Thank you. How are arguments weighed in the computational argumentation? Are they weighed equally? If you have one argument that's for and one argument that's against, but are they equally? Are they equal in the sense of arguments?
They don't have to be weighted equally. That's one of the things that the user can tune. And one of the things when I mentioned we look at sort of the source of the information, you might say, well, here's 2 sources and there's one source that I think is more reliable. So I might trust the information from that source more than information from another source. So then you can temper the weight of an argument based on what that source is. It's the user can do that balancing.
So the user defines reliability?
So the user can tweak it, yeah. So, yeah.
And is it like, are users also, so then the next question is, are users, how are users weighed? Are you weighing users? Is one user more important than the other user?
We're not weighing users.
Okay.
Users can see what the information is. So they would see, let's see if I can go back. So like this, they would be able to see this kind of information. We have different ways of showing this. And the full database is much bigger and more complex than this slide. But you could, as a user, drill down and oops, sorry, wrong button. You could read these different things and say, okay, well, actually, I don't agree with this piece of evidence, so I'm going to reduce the importance of that one. Or you might just say, well, I know the source of each of these arguments, and this source I think is really reliable, and this source I think is a bit sketchy. So I'm going to take everything from this source and weight it less than everything from this source, for example. So, different users have different levels of tolerance for how much detail they want to drill down into. And so the idea is that because there's no single right answer, you're going to get different preferences from different people in terms of whether the kind of thing that they're looking for, you want them to start with something that they know the answer to themselves so that they can basically effectively train the system, not in the machine learning way, but train the system to be giving answers that they feel comfortable with and that they rely on, so that when they get new information, then they can feel comfortable with that answer that's provided. Okay.
Thank you for the presentation. I have two questions. Number one is, how much of foundational data set is needed to reproduce this? Then number two, do you have consideration for simulation, like you deriving crop improvement parameters for simulation to improve the system?
So the first question about how much engineering is required. So in terms of knowledge engineering, you need a fair amount of knowledge engineering because we start with an empty system. And this is the first time we've applied computational argumentation to plant phenotyping. Now, if we move on to another project, suppose we get a project funded about wheat, which is another big crop in the UK, we could look back at what are the things from miscanthus that seem relevant and port those and be sort of like a transfer learning kind of concept where we'd say, well, what are the things that we learned from the miscanthus project that would carry over to wheat? And then what are the things that are irrelevant and we get rid of them? So you wouldn't have to start completely from zero. But there is a fairly substantial knowledge engineering aspect to it. Sorry, I forgot your second question.
My second question is there any
simulation?
Computer simulation, deriving crop improvement parameters to be fed into simulation?
Right. So sort of a longer answer to that, but I'll start by kind of saying, well, there's visualization and there's simulation. So what we're trying to do with this, I come back to here. With this, we're trying to basically build a full 3D reconstruction of visualization of what's currently in the field. So we're not trying to predict anything. We're just trying to build a representation of what's there. Okay. Now, from sort of where I come from, simulation implies being able to kind of forward predict what might happen next. And we're not looking at this with the current project, but you could think about, well, if I'm building, because obviously we're taking data multiple times a year, so we could build a time series of what happens over time, and we could take this representation of, how this was, this was taken last week. So how was this image, what's going to happen with that in 6 months, and then simulate that forward. We're not doing that at the moment, but you could do that. So, I think what we want to be able to get to at this point is something where this, which I said, like, it looks kind of crude, and we want to be able to get this so that you could imagine as a plant breeder being able to zoom in. This is just a screenshot from a program that'll give you sort of 3D visualization where you can zoom in and out and you can pan around. And if you're a plant breeder and you're spending days and days, especially in the UK where it's cold and dark and rainy and wet, and you don't want to be going out to the field all the time, if you can get some images like this that'll save you some trips out to the field, then it will be a useful thing. So can we get the fidelity of that good enough so that they feel confident that they can go out there? That would be a first step. So I want to get to that. I want to be able to visualize what we're doing, what we see now, before trying to then go, we can forward predict that. We have the technology to forward predict it, but I think we need to get to a better trust level here. Okay.
So if you have more questions, please find Dr. Sklar during the coffee break. And please join me in thanking Dr. Sklar for her great talk. And we will reconvene this session for the remaining talk in the same room at 3:40. See you then. Thank you.